Dr. Dora Hermes is our speaker today. She studies the signals measured in the living human brain in order to identify biomarkers of neurological and neuropsychiatric diseases and to develop neuroprosthetics to interface with the brain. She did her Ph.D. working with Nick Ramsey at the UMC Utrecht in the Netherlands, where her work on integrating functional MRI with intracranial EEG measurements received the Editor's Choice Award from the Organization for Human Brain Mapping. During her postdoc, she worked with Jonathan Winawer at MIU and Brian Wandell at Stanford, and received a prestigious Veni-fellowship in the Netherlands to study the relation between visual input and neural oscillations in health and disease. She is now an assistant professor at the Department of Physiology and Biomedical Engineering at the Mayo Clinic in Rochester, Minnesota. She's almost been here a year, and she's on my thesis committee, and I'm really excited to hear her speak. So without further ado, Dora.
So thank you, Tori, for the very nice introduction. Thank you so much for having me here in a virtual setting. And today I would like to tell you a little bit about our work on visual encoding models of human field potential recordings. So to start out with a picture of some of the things we may see in our environment. And these are pictures, and the first thing that may attract you in these pictures that you may look at are these two kids that are my two kids, which I think, of course, are very cute. And there's objects like benches or a bed or a shirt, etc. But what I actually want you to pay attention to in these pictures are patterns. So bars and checkerboards are very common patterns in our environment. And did you know that these type of grading patterns can actually trigger seizures in photosensitive epilepsy, if it were larger than this example? So in photosensitive epilepsy, it may be well known that flashes or flashing lights, such as a stroboscope, may cause seizures in patients that are sensitive or people that are sensitive to that. But patterns, high contrast gradings, can also trigger seizures in a subpopulation of these patients. And moreover, these type of patterns can cause headaches in people with migraines, or may just simply, if you see a large pattern of stripe, may cause some discomfort or some illusions of flickering in normal human beings. So these, for normal people, it may be just a little bit of discomfort when you see a grading, but it can have a large impact on photosensitive epilepsy. For example, there are families who have little children that have Dravet syndrome who are very photosensitive. And for 2 years, a family with a child with Dravet syndrome, they only went to the playground at night because during the day, the patterns at the playground were too bright and too provocative and would cause seizures. Another family, because of the high contrast images and provocative environment in the city. Other people change the interior of their house because of radiators or blinds can trigger seizures. So understanding the computations from various visual input to typical and atypical brain signals is essential in terms of to understand why these patterns can be so provocative in photosensitive epilepsy, or simply causing discomfort in normal human beings, or why they might, cause headaches in people with migraines. So what I want to show you today is I want to show you something about encoding models of human field potential recordings. And encoding models work with visual input, such as patterns or objects. And then, you have a brain that processes these visual inputs. On the left, there's the front of the brain. On the back, there's a back. And in color, I have highlighted some of the areas that are involved in visual processing. In red, basically V1, the first visual area, cortical visual area that processes images and visual inputs. And then you can see a field potential signal coming off of that area. And those signals have different features that may tell you something different about what's going on in the brain and what type of feature in the image is being processed. And encoding models simulate a circuit and try and predict these brain signals by using canonical approaches, canonical calculations such as filtering, rectification, or summation. So the outline is first I want to show you how the stimulus dependence of field potential signals in human visual cortex, such that we can start seeing how they depend on images and which signals are particularly driven by the same signals that also cause seizures in photosensitive epilepsy. So we want to see how different patterns and inputs drive the visual signals. Then I want to show how we link the stimulus dependence of field potentials of photosensitive epilepsy. And then lastly, I want to show you an image-computable model, so an encoding model of field potential oscillations in the gamma range. And in order to measure field potentials in the human brain, I measure intracranial EEG signals in humans. So intracranial EEG is a standard clinical procedure for the treatment of pharmacoresistant epilepsy. And when patients have these electrodes implanted for epilepsy monitoring, we have IRB approval to ask these patients whether they want to participate in behavioral tasks during their clinical stay. So you can see here that this patient has electrodes implanted and the neurologists are trying to localize the seizure focus. And you can see that they're implanted on many areas from the middle of the brain that has motor cortex, from the back here of the brain that has visual cortex highlighted in colors again. And you can see that there's many electrodes implanted on many different areas. And that's because neurologists have several hypotheses about where the seizures may come from, but they are not sure. So they want to know, they implant electrodes on areas where they think the seizures may start, and they want to localize that very accurately and only resect a small area that may be involved in the seizure productions and leave the rest of the normal functional brain completely intact. So in these patients, there's often a small seizure focus, but the rest of the brain is functioning normally. Also, these patients with epilepsy generally do not have photosensitive epilepsy, but different types of epilepsy. So we use this as a model to understand typical field potential signals in humans. And these field potentials have many different features that reflect different underlying processes. And one of these features has been highlighted since the 1980s, and that's gamma oscillations between 30 and 80 hertz. And a gamma oscillation, an oscillation is really like a regular time scale. You can see that there's an oscillation here, sinusoid, which has a peak in the power spectrum. So if we think about the oscillation and you have frequency on the x-axis and the log power or power on the y-axis, the power spectral density shows a peak at a particular frequency of the oscillation. And that indicates that the circuit underneath is doing something in synchrony. And this can be generated, for example, by a loop between the excitatory and inhibitory neurons. And there have been many hypotheses about the role of gamma oscillations in human brain function. For example, gamma oscillations have been proposed to be essential for perception, for conscious perception, or for feed-forward processing. However, there are other signals as well, and much later, in around 2007, people started to pay more attention to broadband signals in the field potential. So broadband signal is basically a random walk. And the power spectrum has a 1/f shape that spans many different frequencies. And if the power in a broadband signal increases or decreases, that means that a lot of different neurons are doing different things. And it has been related with models and with measurements to Poisson input firing rates. So random input firing rates that get integrated in the neurons and then produce a 1/f power spectrum. So the first is an oscillation related to neuronal synchrony, with many hypotheses about how that's essential for brain function. The second, the broadband signals, are simply a measurement of the underlying asynchronous neuronal activity. When I started using ECOG recordings in the human brain, we knew of these gamma studies in macaque, and you can see here one of the power spectra with here that was recorded in macaque with the frequency on the x-axis and the power on the y-axis, and you can see here that during several visual stimulus presentations, you can see this very clear narrow band gamma oscillation around 50 hertz, and then you can see these broadband increases that span many different frequencies. However, when we measured the spectral power changes, as you see in green on the top during finger movement, or in temporal higher order visual areas during face and house processing, we didn't see any of these gamma oscillations. We only saw these broadband signals. And when we were doing these studies, we would get many questions like, is this broadband just gamma in ECOG? But it's a very different phenomenon. So we didn't think so. So why do we not see these gamma oscillations in human ECOG recordings? Well, they have been proposed to be essential for perception. So why do we not see them? So we went back to the literature and we looked at a lot of the different studies that had looked at gamma oscillations in mostly macaque research, and they all used gradings or striped patterns. And this has a historical background because gradings and stripes drive visual cortex strongly. It's been shown since Jubel and Wiesel that early visual cortex responds to oriented stripes. So if you have a stripe pattern that drives visual cortex in a very particular way, and if you want to record individual cells showing a bar on the screen, an oriented bar of the right orientation, that will drive that particular neuron very strongly. So we pulled out some gradings and we had recordings in early visual cortex where a patient had electrodes implanted in the back of the brain. And this is a raw signal of the intracranial field potential, and you can see a very nice, this early peak is an evoked potential, and then after that you see this very clear gamma oscillation. And then in the power spectrum, you can also see that there is a clear peak in the power spectrum at around 50 Hertz. So we found our gamma oscillations for this grading stimuli. And you can also see in blue that this stimulus also induced some broadband increases. However, when these subjects looked at noise patterns, such as you can see here, you see an evoked potential as well, but then you see more of a random walk. We do not see that oscillation, and we only see a broadband increase in the field potential, even though in both cases, the visual cortex is processing information. It's in a very different, the circuit shows a very different dynamic. So if we look at all the different gradings that we showed, you can see these are 2 example electrodes, but this was reproduced across all the electrodes in V1 and V2 and V3 that we had in these studies, in these 2 subjects. You can see that there's a very clear gamma oscillation for the grading images, whereas all the images show a broadband elevation in power. So gamma oscillations are highly stimulus dependent and induced by one particular stimulus and not by others. So we demonstrated the stimulus dependence of gamma oscillations, and we hypothesized that gamma oscillations are a biomarker of a very particular circuit dynamic in visual cortex, since they are driven by grading stimuli but not by noise patterns. And both contain contrast, and whereas in the broadband signals we saw increases, changes with spatial frequency of the noise and the gradings that we were expecting from visual cortex, the gamma oscillations were basically there for the gradings and not for the noise patterns, but noise patterns also induce firing in early visual cortex. So broadband signals are a general biomarker of visual cortex activity. So after demonstrating how these 2 different signals depend on the visual input and the stimulus dependence of gamma oscillations, I was talking to some of my colleagues in neurology about these results, and that's when we started linking the stimulus dependence of field potentials with photosensitive epilepsy. So as I just mentioned, grading patterns can trigger seizures in photosensitive epilepsy, and we showed that gradings induce gamma oscillations. So what we wanted to do is we wanted to compare the stimulus features that induce photosensitive epilepsy and that induce gamma oscillations. So we went back to the literature, and people have studied the different stimulus properties that drive gamma oscillations for a long time, and so over the last few decades now. And also in neurology, people have also started to test which stimulus features can induce seizures. And what I just showed you is that noise, if you increase noise, then basically gamma oscillations go away. And similarly, epileptiform responses, the literature shows that epileptiform responses also go away when you increase noise on top of a grating. And also, we compared many other features, size, so increasing the size of gratings, decreasing contrast, increasing the number of orientations, changing the luminance from black and white to red-green chromatic, increasing spatial frequency from low to middle, middle to high, changing the rating edge. Gamma oscillations change in a particular manner, and epileptiform responses change in the exact same way. So whereas a larger grading, for example, increases gamma oscillations, it also increases the number of epileptiform responses measured in patients with photosensitive epilepsy in the EEG. However, firing rates change in a very different manner. So when you increase the size of a stimulus, for example, firing rates go down due to surround suppression. Also, firing rates go up or stay the same when you increase noise on a grating in V1, whereas gamma oscillations basically reduced. And so you can see that also when we look at the fMRI response, it's very different. So it's really the same type of features that drive epileptiform responses also increase gamma oscillations, and therefore we hypothesized that gamma oscillations and epileptiform responses in photosensitive epilepsy are driven by the same circuit. So we linked the stimulus dependence of field potentials and photosensitive epilepsy. So now that we know that the same are linked, we can start leveraging our knowledge of gamma oscillations to develop models that predict gamma oscillations and potentially also which stimuli can cause seizures in photosensitive epilepsy. So we wanted to develop an image-computable model of field potential oscillations in the gamma range. Now, image-computable models, or this type of encoding models, can have very many different forms. And we wanted, you can have like a very naive approach, for example, where you train a deep neural network to approach the same aspects of the circuit. Or you can use the knowledge of the system to gain insight into computations rather than that. So, and that's what we chose. And you can also use many different training image sets. You can use natural images, but we chose to use a relatively large set of 86 patterns with well-defined image features to train our model. And I'll come back to why I think that these two choices helped us afterwards for the goals that we were trying to reach. So we wanted to develop an image-computable encoding model. And therefore, we built on previous developed image-computable encoding models that were developed for fMRI. And I'll show you a model that was developed by a collaborator of mine, by Kendrick Kay and others, of an image-computable encoding model that explained fMRI bolt signals. So these kind of models simulate the network to predict a response from a stimulus. So what does that look like? This is the second-order contrast model from Kendrick Kay. So images get filtered with, in this case, with Gabor patches, rectified and integrated, and then you get oriented contrast energy image. So how much contrast is there in all these 8 different orientations? So we calculate how much diagonal, how much horizontal, how much vertical energy is there for each of these orientations. And then there's the process of divisive normalization and summation across orientations in this model, so which results in a contrast image, which indicates how much contrast there is in many orientations in different parts of the image. And then this model that explains the fMRI signal well has a computation that is variance across space, which basically indicates how much variance there is in how much contrast there is in different patches. And then the signal may be larger if the contrast varies a lot. So if there's interesting patterns that are not very uniform, like a grading, the signal may increase. And then there's just a power law nonlinearity to predict the fMRI responses. So, and our stimuli, so that's what we started with. We started with this image-computable model for fMRI, and we used the same images that were used in these fMRI models. And here you can see some of these examples. So from left to right, the images contain these squiggly line patterns that varied over space from left to right and from bottom to top, because different areas of the visual cortex prefer contrast in different parts of the visual field. The left responds to the right responds to the left, top responds to the bottom responds to the top, etc. Gradings of different orientation, increases in contrast for gradings plaids and a pattern with 8 orientation, and increases in contrast for these squiggly patterns, then changes in sparseness, coherence, and noise. And then one of the things is when we showed these images to the patients, we wanted to decompose the power spectra, as I showed before, into broadband and narrowband gamma components. Broadband signals and narrowband gamma signals. And you can see here an example of how we fit the data. You can see in black, you can see the power spectrum. Here's frequency, here's power, and in black there's a power spectrum during rest. In blue here, you can see the power spectrum during one of the noise patterns from these 86 images. And you can see that it's well fit by this dotted line, which is just captures the broadband component. And that's the same for all these example electrodes in these 3 subjects. If we look at the power spectrum in red for the grading images, and these 2 lines just indicate the confidence interval, you can see that it's well captured by us fitting this straight line plus a Gaussian, which captures the narrowband gamma components. And it's very essential to fit a model like this, because now we can take this broadband signal plus a gamma signal and explain each of them separately and tease that apart for every single image. So we had our 86 images, and there were already existing fMRI data for every one of these images, where you can see that the first image didn't show a large fMRI response, so every bar represents the response to one image. And for example, the fMRI signal in this example voxel likes images on the right, doesn't like contrast on the bottom, likes contrast on the top, has a larger signal for these squiggly line patterns compared to the gradings, and then increases in signal with increasing contrast. Here especially you can see a nice increase in contrast across these squiggly lines increasing in contrast, and then has a smaller signal again for these striped patterns. And we can see that when we take the ECOG signals from these patients that I just showed you, and we extract a broadband component, we can see that it matches very well with the fMRI signal, and we can use the model that was developed for fMRI to predict these signals. You can see that ECOG broadband encodes similar features as fMRI. You see that the signals are, in this case, smaller on the left compared to the right, because we match these 2 areas of visual cortex. And then also the responses to these patterns are larger compared to the gradings, and then there's an increasing response with contrast. However, when we look at the narrowband gamma component, it looks very different. There's barely a signal here for these images, and there's a large signal for the grading. So we needed a very different image computable model. So we had to develop a different image computable model of gamma oscillations. It had to be different than the one that explained the broadband features, because when we tried to fit the broadband, the same model as before, we didn't explain a large percentage of our signals. So our model was similar. So we filtered the images and similarly computed oriented contrast energy images, but rather than summing across all these images as was done before in the model for fMRI, we sum each of those immediately within the population receptive field to which the electrode is sensitive, such that we get 8 values for how much contrast is there within one particular orientation. And then we calculate the variance, which is across these 8 different contrast areas, which basically is an indication for orientation homogeneity. So how homogeneous is the part of the image that drives the part of visual cortex? It's very homogeneous. It's basically a very, in terms of orientation, so has a single orientation present, such as in the grading, it will strongly drive this model. If there's many orientations present, then it will not drive this model at all. addition to that, if we have higher contrast, then this value will automatically be larger. If there's a single, only if there's a single orientation present or few orientations that are present. So when we fit this model on the narrowband gamma signals in these three across the electrodes in these three subjects, we explain a mean of 75% of the variance. So which was really nice, and we can see that it tracks the features well. You can see that it's large for these and it increases with contrast, like more lines rather than less lines, etc. So we wanted to have a model that used the knowledge of the system to gain insight into the computations rather than a deep neural network. And this model taught us that contrast, spatial extent, and limited orientations all predict larger outputs in the gamma model. And the nice thing is that we have that we can now say that these different computations have all been associated with gain control or suppression in neuronal circuits. So we therefore hypothesize that gamma oscillations are related to gain control rather than simple feed-forward processing. And if we go back at how can this be implemented in a circuit in visual cortex, there have been several of gamma oscillations. There have been several models, several circuit models, such as the one on the left with an E and an I component, an excitatory and an inhibitory component that models a cortical column in visual cortex and receives input from earlier areas. And then there have been later iterations of this model where there is a global component or a surround inputs to the specifically to the inhibitory components that may play a role in stabilizing the net responses for the network for large gratings. For example, homogeneous orientations. So if we now have, so when we know these kind of features, we can start thinking about how these global components that were necessary in these models, how those are related to the homogeneous orientations in our image-computable model, and think about how that how changes in these network dynamics can induce epilepsy. For example, when these surround inputs are disturbed, would that then destabilize the network response? So we can see that these typical features are all associated with gain control or suppression in neuronal circuits and have been implemented in models that we just saw before, where these features stabilize and are really there to stabilize a network. Then the second question that I asked is why was this image training set beneficial? Would it have worked? Like, we had a relatively large set of 86 well-defined patterns, but could we have used a natural image set instead? And to train models, you need a large variance in the response. So if different images drive different responses very strongly, yes, you can. That's what you would expect. Maybe natural images would have induced even larger responses. So what we did to test that, what would be our model predictions for natural images? So what I'm first showing here is on the y-axis, you can see the gamma prediction for the new model, and on the x-axis, you can see the broadband prediction. So you can see that for gratings of increasing contrast, this is the grating with the lowest contrast, predicts low gamma, low broadband. But if you increase contrast, 100% contrast gratings, our models predict a large gamma response and a large broadband response. And we can look at two natural images, and we view these images through a particular electrode that we included in our model. And we use the parameters fit for this electrode. We can see that it's an electrode that responds to the top right of the image. And that when we feed these image features, when we filter these images and feed these features into our model, you can see that it does predict, these image patches predict a large broadband response, but a very little gamma response. So if we look at many natural images from a standard image bank, they actually do not predict large gamma responses. So if we would have used this image bank and only had measurements in this electrode, we could actually not have fitted our model very well, because we just don't have that difference we don't have the variance to predict our model, to fit our model. So you have to be very specific about what types of image set is used to predict this model. And what you can also see is that grading drives the circuit in a very non-natural manner. Large grading batches are not necessarily present in the common environment, especially natural images with flowers. And images need to span a large feature space to train these type of models. So we've seen how the stimulus dependence of field potentials in visual cortex, with many images driving broadband signals, but only a few selective images driving gamma oscillations, and how we've linked that to photosensitive epilepsy, that the same features that drive gamma oscillations also drive photosensitive epilepsy. And we developed an image-computable model of these field potentials that predicts these field potentials in the gamma range. And what we can now do is that we can start implementing such image-computable models, for example, in a smartphone app. So thanks to the software gaming project from the University of Utrecht, I worked with a group of CS students there, and you can see here the screen of a smartphone, and in every square or every rectangle here, you can see a number. And so this app filters the images, input image in real time, doesn't take color into account, only, and is most sensitive to gradings of about 3 to 4 cycles per degree, which is the same frequency that drives gamma oscillations very strongly, and also is most provocative in photosensitive epilepsy. And in real time, it filters the input and it predicts what the output in the model is going to be. And if you see, you can see the phone moving around the environment, and you can see that some image patches, such as right here in the middle, strongly drive gamma oscillations. And for example, escalators that are well known to potentially be provocative in photosensitive epilepsy and sometimes disturb people with migraines are also highly provocative. And then you can see that other patterns in the environment, such as this kind of radiator, also strongly drives the model. So we can start implementing these type of models in different type of applications. So, so in terms of, you may wonder, like, can you reproduce these results? Are these results something that I can build on? I would say yes. So for all the publications that I showed, our code and most of the data that were essential for making all the figures in the paper are shared on GitHub, and data are also shared on the Open Sciences Framework. And there's code to generate all the figures, to download the data and generate the figures, which are in the research style, not official style packages. For example, script 01, figure 1 should make figure 1 of the paper. So for all these 3 papers, you should be able to reproduce the entire results based on these. And in order to facilitate reproducibility and reusability and interpretability in my lab, we try to work in a very systematic way and store our data now in the, we've been working with the brain imaging data structure. So the brain imaging data structure is a community effort towards a shared structure for neuroimaging data, which allows for easy multimodal integration. If it's used, for example, for both fMRI and intracranial EEG and EEG, we can start building models for each of these signals using machine-readable and human-readable metadata. And it allows for automated computation. So we extended the BIDS IEG, the BIDS structure that was initially developed for MRI to intracranial EEG, which was just published last year. So now we can start making models from neuronal populations to most of these different signals. EEG, MEG, MRI, IEG have all been integrated now, and multi-electrode recordings are still an ongoing effort in BIDS to see whether researchers can store the data in the same way. So to summarize what I've shown you today, we are studying separate components of the ECOG power spectrum And frequency ranges do not necessarily separate these different components, and we had to different processes, but we had to fit like this broadband component and this gamma component separately to be able to predict these results reliably. So different features encode different visual stimulus properties and different properties of the circuit. And the same image features that drive gamma oscillations also drive seizures in photosensitive epilepsy. And once we know these things, we can make models that provide quantitative predictions. And we can further test and refine these models and see where they're wrong. It's a model. It's not an accurate prediction of the circuit. But we have to start somewhere to start making, to be able to make these predictions and test where they are wrong. And then we showed that ECOG gamma oscillations could be predicted by variance across orientations or orientation homogeneity, and that they may be a biomarker for gain control rather than a signal that quantifies feed-forward processing. And it's important to share code and data to ensure reproducible science and reusability of these results. So I would like to thank you very much for your time, and I would like to thank my collaborators, Kai Miller at Mayo Clinic, Natalia, Nick, and Dorothy at UMC Utrecht, and John Winawer at NYU, Kendrick Kay at University of Minnesota, and Brian Wandell at Stanford, and of course my lab here, and the amazing intracranial EEG teams who have all helped a lot in the data collection for these studies. And thanks to my funding. So I'm happy to take any questions.
So that video that you showed with the camera like panning and giving different scores for stuff that would be more likely to cause seizures, I guess. Is there any utility for that or any way to like implement that for a family who has a kid with that type of epilepsy?
We are currently in the process of thinking about how, whether patients would find that useful or not. So this is really a prototype to now take to the next level to see whether that can start, to what extent different patient populations, such as migraine, for example, migraine patients who are moving, who have an office environment they would like to screen, or whether parents of patients with epilepsy would find this useful. So now we're starting up studies to see which type of patient populations could potentially benefit from these types. So I also see a question in the chat window. How the advancement of knowledge of neural oscillations can help us improve the technology of diagnosing and treating epilepsy. So when we start to understand the role of neuronal oscillations and implement these models, we can specifically change these models and see what type of how the models would then respond to different image features. So, for example, if normally we would predict an increase, an increase with contrast, that has a certain suppression for higher contrast. But if it's linear for certain types of epilepsy and then results in a seizure, then we can see which model parameters of the gamma oscillations would produce these results. If there's, for example, a change in different frequencies in different patient populations for different stimulus features, we can start using our model to predict like what type of image features would predict different frequencies in gamma oscillations. So we can, if we have these models, we can start maybe separating different populations that might respond to different treatments. Any other questions?
Well, Dora, thank you very much. A great presentation, very interesting. And it's how do you find, just maybe a quick one wrap up, you find a difference between medicine in the Netherlands versus over here in the United States?
In terms of the epilepsy programs that I've been involved in the Netherlands and here, it's actually surprisingly similar. The group in Utrecht has, who I worked with there, has very highly trained epilepsy neurologists where the patients are in the ICU and they're monitored in a very consistent manner. And it's actually very similar as here at Mayo Clinic. there's so I think in terms of the environment that I've seen, which is a very small part of medicine in terms of these studies, it's a very this is very consistent in terms of the monitoring that they do and how they monitor these patients.
Thank you.
I see another question popping up in the chat window. Is the end goal to avoid stimulus or to operate on the brain to reduce the impact of the stimulus? There's two. So the first, so this is would depend on the patient. Many patients benefit, and that's the first thing that's on the epilepsy foundations that I've heard from neurologists. Is the first thing that they tell patients is to avoid stimuli that might potentially be provocative. So for example, if people are highly sensitive to striped, high contrast stripe patterns, don't put those blinds in the room, for example. So just the first thing is, or if you haven't slept for a couple of days, don't look at flashing lights. So that's the first thing that I've heard from neurologists that they tell their patients. However, when that doesn't work anymore, then it's a different story. But because if, for example, in Dravet syndrome, where the brain is very excitable, then we would want to develop new technologies to reduce the excitability of the brain. So in that case, for those type of approaches, we are starting to study different types of electrical stimulation to see whether we can reduce the excitability of the brain. And then we may use these types of stimuli as a biomarker to see whether the brain is actually less excitable, or how different types of treatment, whether these are really biomarkers for excitability of visual cortex. So it depends on the type of patient population that we would see what the goal would be.
Regarding the excitability, is that a conscious? I mean, do you know that your brain is being excited that way, or is it subconscious in a odd way? I know nothing of epilepsy, so.
So it's so if you don't have epilepsy, you can look up a large striped pattern, and you can see these kind of sometimes these patterns can kind of dance a little bit, or they dance a little bit in front of your eyes, or they flicker a little bit. That's called pattern glare. If you look at pattern glare, you can also see that that can happen. So that means that it's a stimulus. It's just a very strong drive of early visual areas, so that's kind of the subjective experience that we have if our brain is driven very strongly. But in terms of excitability, some people are aware of the fact that they have epileptiform activity and others are not. So it depends on the patient population again. Some patients, like migraine patients, want to avoid these patterns at all costs, whereas like some really severe types of epilepsy that are often comorbid with certain types of autism, it can happen that they seek out these patterns to look at them and then move their eyes to kind of induce these features. So it really depends on there's a large diversity, but it happens in many different clinical syndromes. So it's, so some people notice and they dislike it, other people notice, other patient populations notice and they look for it. And as normals, some people might find a building with stripes very nice to look at. Other people may find it causing discomfort. So there's a large variety in how people respond to these type of stimuli.
Sounds like there's no more questions. Is there any more in the chat box that you see, Dora?
No, I don't see anything else in the chat box.
Okay, well, thank you very much. Tori, did you have any closing words?
Nothing for me, just thanks for Dora.
Yeah, thank you very much for having me. That was thank you very much for the questions.
Okay, thank you very much. This concludes the lecture for tonight, and thank you for joining us, everybody. Have a great evening.
Thank you very much. Have a good evening.